This is a video to show the floating leaf disc assay for photosynthesis. Here's the basic setup that each student group is going to need. It shows you each of the materials that they will require.
So what I have on the document camera is our basic setup. Now I'm going to mess up my story a little bit because we're going to be using plants. Because they're easier to use than cyanobacteria, honestly. Okay, so today our subject of choice is spinach leaves, and what we're going to be doing is first of all making some small discs of these leaves. So in order to do this, I'm just going to put it on the doc cam so you guys can see, we're going to use hole punches, and I have several in the room today that we can use. Now when you make your discs, you want to punch them out so you get nice even circular discs, just like that, yes? And you want to, as much as possible, avoid the major leaf veins. So just to be clear, you see this right here? We can even zoom in so you can see this a little better. The main veins in the leaves do not have as much chlorophyll as other cells, so we want to avoid those as much as possible. And I want to just go ahead and punch out, your directions call you for punching out 10 discs. And I'm not going to punch out a whole bunch, I'm just going to punch out a few. You guys can punch out the 10 that you need. Okay, now the issue with these discs is they actually have some gas present in their tissues now, and I don't want them because that's going to affect the results of my experiment. So right now I want to take a second and get rid of any gas that's present in the tissue. So I'm going to take a syringe without the needle, okay, and I'm going to first pull out the plunger, just like that. I'm going to pick up my discs and put them right into the syringe. Now, you have 2 syringes on your bench. You're going to be working in partners. So you and your partner are going to do this in parallel. One of you is going to work with one syringe. One of you works with the other. Okay? Add the plunger. Now, all of your directions tell you how to do this. I just want to show you because I feel like the directions are a little bit wordy, and I think this will help us. So go ahead and depress your plunger. Now, don't schmoosh, technical term, your discs. Okay? We don't want to damage them. That will definitely affect the experiment, yes? So what I have in the front is just a cup of regular water. I'm going to pull back on my plunger and drop some of that water. So good? Okay? Now, just like you would if you were a doctor, you're going to go ahead and get rid of any air present in that syringe. So as you do this, they are not water guns today, gentlemen. Do not aim them at someone and start spitting them out.
Spinach water.
So we want to be sure to flick the syringe, spit out any excess air. See, from this, they're floating, right? Why are they floating? There's air in the tissue and they are less dense than the water. Nice. So I want to suck out all the air. So to do that, I'm going to put one finger over the top of my syringe and I'm going to pull back on my plunger, creating a vacuum. Now, I let go of the plunger and then I let go of this, and what will happen? What's starting to happen, guys?
They're swimming.
They're starting to sink. Why are they sinking? I sucked out all the air. Now, wait a minute. I have a couple there at the top that are still floating. I can flick it. Where did the air go? How did that happen, guys? When you put something in a vacuum, I could do this again just in case I'm worried that they're not all sinking. Right? You pulled it out of the tissue, and look, do you see an air bubble that wasn't there before? Yeah. That came from them. So I want to do this until all of my discs are sinking. Ahead and add them to this cup. Now you notice I have 2 cups, right? And I told you you have 2 partners. One of you has one of these syringes. The other one is doing exactly the same thing, like you're twins, yes? Okay? So I'm going to add my cells straight into these cups. Before I do that, one of these I'm just going to leave it alone. Just going to be water. It's going to have these discs in it. What am I going to call that? The control. The other one I'm going to add some magical baking soda. So you're going to add about a pinch. You're not going to weigh it out. You're not going to measure it out. You're just going to be like you're cooking. Bam. Just like that. Right? Just like that. Now, when you take off your plunger, you're going to pull it out. Obviously, it's going to leak. It'll leak right into the cup. Not a big deal. And you're just going to pour your discs right into the cup. Now, realizing I will have discs in this cup as well.